welcome everybody. Good morning, good afternoon, good evening, wherever you are, and thank you for joining. So this is the first webinar planned by the HTA ESIG as part of EFSPI, which is the European Federation of Statisticians in the Pharmaceutical Industry. So this is what we're hoping to be the first of a series of webinars. Briefly a little bit about me, and then I'll tell you a little bit about the ESIG, and then I'll hand over to Anna, who can get us started. So my name's Ash Bullerman. I'm one of the directors in the HEOR team at Petauri Evidence, which is a consultancy company, and I'm also a member of the HTA ESIG, and I head up our training sub-team. So within the HTA ESIG, we have members from the pharmaceutical industry more broadly, including people working in industry, in pharmaceutical companies, in consultancies, at universities and other organisations. More broadly for EFSPI, the organisation has a lot of different research interest areas and special interest groups. We are a European special interest group looking at HTA. Our goal broadly as the ESIG is to bring together a group of people who are statisticians working in the pharmaceutical industry that have this shared understanding, interest in advancing methods and how we do statistics for HTA purposes. We have a couple of sort of key goals that we're aspiring to work towards, and those are listed on this slide. We're trying to empower statisticians who work in HTA through this platform that we have as an ESIG to share and co-develop best practices. We do that sometimes through working papers, presentations at congresses and that kind of thing. We try our best to foster interdisciplinary and intradisciplinary scientific dialogue on HTA statistical matters. A lot of that more recently has included discussions around JCA, as I'm sure people are very familiar with. And we try and use our collective expertise in HTA to provide, hopefully, useful but influential input to policymaking. And so that's where we've commented as an organisation on various methodological processes and policies that have come through for HTA agencies and then broader organisations like the European Joint Clinical Assessment documentation. And this last point I've put in bold because that's why we're here today. So it's to promote education and awareness about the importance and applications of HTA statistics. And that's what we're hoping these webinars can be useful for helping us do that. So if you want to learn a bit more about the ESIG and who we are and what we do, there's a QR code that takes you to our website. And there's also an email if you want to ask any specific questions. So that's a little bit about me and the ESIG. Just a tiny bit of housekeeping. So the plan today is to run until the hour. We know people probably have meetings, so we'll make sure we're done by then. We've very helpfully had a lot of questions submitted in advance. And as an ESIG, we've collated questions that our members have shared with us. And that's really been incredibly helpful for us to shape this webinar into something that we hope is as useful as possible. So you'll find on this software program, there is a question box, or it might just say questions. You are more than welcome to post additional questions in there, and we will try our best to get to them, but I will not promise that we will, because we've had quite a lot of questions in advance. But please do put them in there if you'd like. And then next to that, at least on my device, there is an option for materials. You can then download a copy of the slides that Anna's going to talk us through in case that's helpful for you. So I think without any further ado, we'll get started. Anna's going to perhaps first introduce herself, and we're delighted to have her to do our first webinar. And we'll go through Anna's presentation, and then we'll move to the Q&A, excuse me, when she's finished. So thank you, Anna. I'll let you get started.
Thank you, Ash. Yeah, it's great to be here. And yes, so I will be speaking today about the introduction and software implementation of non-proportional hazards in network meta-analysis, and in a little bit more detail, fractional polynomial and piecewise exponential model. And yeah, so I work as a associate director biostatistics at BMS in Switzerland, and currently I'm working in the HTA department supporting our HTA statistics. Yes, but with that I will move into the presentation. And I would like to really acknowledge the HTA ESIG training sub-team, and especially Ash Pullman and Christina Daskapolo, who provided a lot of good insights and ideas how to build this. And then disclaimer: so all views presented are mine and not necessarily the affiliated organizations. And then the content of the slide deck is the accurate to the best of my knowledge at the time of production. Okay, but then let's go to the topic of today. And it's always good to start with why have the models for non-proportional hazards become more common in network meta-analysis. And I have here this figure from a paper, Statistical Issues and Challenges in Immuno-Oncology, which shows different survival curves. So we have there A, proportional hazards, exponential model. Everything is nice and green, and you can fit the proportional hazards model. But then we could also have a proportional hazards with long-term survival. And then in the C, there is a delayed effect, clinical effect, and I think this was especially something which, when the immuno-oncology drugs started to come on market, that there was a delay in the clinical effect, and then of course we could have also long-term and delayed clinical effect together, and then there can be also other types of situation where the non-proportional assumption doesn't hold. But in order to be able to take these into account in network meta-analysis, these methods have been developed. Okay, and then the topic of today, fractional polynomials in network meta-analysis, is something that I became across them 2016, so 10 years ago. When I was reading the great paper by Sharon Jansen, and it seemed quite complicated, and it is a challenging modeling framework, because we have the network meta-analysis, which creates certain modeling things which needs to be taken into account, and then also fractional polynomials, and combining those two, and today I hope that we can kind of get a little bit idea what is behind these models, and understand what they are modeling, and put it into pieces. Then of course nowadays you always should ask if the ChatGPT can do this, and I did that actually already almost a year ago in February 2025, and it gave me a quite long R code and Jax code and so on. But then it also gave some notes, like fractional polynomial models are complex and may require advanced statistical knowledge and custom coding, and consider consulting a statistician. And I was quite happy to see that there was that don't just take this and so on. Then obviously the developments in AI are fast, and I decided to ask again. Now that I was preparing for the webinar, and now I got much more confident response. So it started with certainly, and then I got a piece of R code which I tried running. However, so it was referring to multi-NMA package, but then the function it was giving is not in the package. So obviously this was this was not something that worked, and we still need to be, you know, able to understand these codes and fit the models and so on. Yes. And with that background, then the scope of this training is based on this publication, Non-Proportional Hazards in Network Meta-Analysis Efficient Strategies for Model Building and Analysis. It's together with Sandro Steiger, and Neil Hawkins and Hans-Peter Piefo. And well, if you want to go back to some details, hopefully everything should be in the paper and really read the theoretical background. And also the example we are going through is the same as is presented in the paper. And as a learning targets, I hope that after that we will provide a basic understanding of the parameterization of NMA models and in the way that it's done in our model building paper. And then what is fractional polynomial model and what is piecewise exponential model. And then fitting the models with R and really linking the R code to the parameterization of the and specification of the models. And in the last part, we look a little bit about the model selection. So yes, let's go to the part one. And for me, I mean, how we specify it is in a form of generalized linear model. And in generalized linear model, we have always the likelihood, link function, and linear predictor. And in this case, its likelihood is the binomial likelihood. Link function is complementary log-log link function. And then linear predictor has different parameters for study and treatment effect, as we always have in network meta-analysis, but then also the time-dependent terms. And let's next go to a little bit more details into these things. So we start with the likelihood, and this is so modeling grouped time to event data as binomial data. This is what we do in this analysis. So we have the number of events for given study and treatment in a given time interval, and that has a binomial distribution with certain probability and certain number of trials. And we get the binomial data from the Kaplan-Meier curves, and this is done by reading in Kaplan-Meier curves from publications. Many of you are likely familiar with that you can digitize them, and then using special algorithms you can convert it to the individual survival times. And when this process is done, we group the data, the time to event data to time intervals. So then in the data we will of course have study and treatment information, but then we will have the time interval. So this is from 0 to 2 months to 4 months and 2 to 4 months and so on. Then always the length of the time interval. So in this case it's 2 months. And in each interval the number of subjects at risk. So it's the n, and then the number of events. And obviously for each interval the number of subjects at risk is decreasing, and it's decreasing because of number of events, but also because number of censoring. Yes, and then the second piece of the generalized linear model is the link function. link function, and we can use a complementary log-log link function to model group survival data with the binomial likelihood. And the link function can be derived as I'm showing here, and this is also in the paper. And then we end up having that our binomial likelihood is with the complementary log-log link function to the linear prediction eta j k t, and then we have an offset term which is the logarithm of the length of the time interval. So that's always there, and this is quite old result, already 1978, published by Prentice and Glockner, and this is also used in the other papers about fractional polynomials and so on. Yes, and then let's dive to the last piece, which is the linear predictor. And we would have there the eta j. I mean, if we wouldn't have any time-dependent effects, so we would be just doing proportional hazards NMA. We would have a linear predictor with study and treatment effect, alpha j for study and theta k for treatment. And maybe it's good to mention here also that this is the fixed effect model part. If it's random, then it gets a little bit more complicated. But let's now concentrate on this. And then if we wouldn't be doing a network meta-analysis, but we would be just modeling the hazard function by some fractional polynomials. Then our linear predictor would be just eta t, and we would have the functions for the time-dependent effects, which are in the paper noted by G0 to Gm. And when we put these together, then we will have in our model, in our linear predictor, we will have effects like the time-dependent effects, and we will have those for the studies and for the treatments. And the M is referring to the number of how many different time-dependent parameters we have. So it's if we have second-order fractional polynomial, then the M goes from 0 to 2. If we have first order, then it goes from 0 to 1, and then you can write all of this open. Okay, and then here I'm listing some different linear predictions for the time-dependent effects. So there are 9 different models, and the first 2 are actually proportional hazards. And how you specify those? First one is the basic exponential proportional hazard model, and then the second is proportional hazards with viable baseline survival. There we need to put the treatment-specific time-dependent effects to 0. And then there are 4 fractional polynomial models and 2 piecewise exponential models. And it's always good to have the proportional hazard model as well, because of course we should test that do we have violations from proportional hazards. And sometimes you might not have, and then we pick the proportional hazard model. Okay, and then here I'm just showing how do different time-varying hazard ratios look like. And I have for the example network here the hazard ratios over time for all the 9 models, and in these figures I have always on hazard ratio on log scale is on the y-axis, and then on the x-axis we have time in months, and then we have treatment codes for treatment 1, 3, and 4, and the curves are the hazard ratios against treatment 1, for all these three treatments. And if we look, I mean, proportional hazards, that's the simplest one. So what it means that the hazard ratios are constant over time, and of course then we wouldn't need the time-dependent modeling, but if we fit it, then it's just constant. Then the fractional polynomial models, 4 of them, and we can see that it allows kind of different functional forms depending on the which fractional polynomials we are specifying and which type of functions. So the hazard ratios can really take different shapes over time. And then the last one, which is the piecewise exponential models, and it's the bottom row. So obviously this allows the hazard ratio to change at some points of time, and in the pieces it is constant. So the first one is with one cut point, and then we have 2 models with 2 cut points, and the functions look always like step functions. And then let's go to the part 2, which is fitting the models with R. And here is just a snapshot of the data, which uses the same headers as in the R code, so that we can see how we need to have the data when we move into the modeling. And I have there the study and treatment, well, actually both as numerical and categorical variables or factor variables. Then I have the time point, time delta, number of events, and number at risk. And then if we do the frequentist model fitting with the GLM, here is the R code for that, but let's first look for one example, the kind of the linear predictor. So this is a second order fractional polynomial. We have the eta jkt is log ht, and then we have here we have the study and treatment effects, like the intercepts, and then we have first order time-dependent terms and second order time-dependent terms. So this would be, we saw the couple of slides ago, the sum of the time-dependent study and treatment terms, and this is then writing it open for one example. And when we go to the model fitting, it's the GLM function in R, and we can just give the outcome, so number of events, and then we will have in the linear predictor the treatment and study specific events. And here is the first order fractional polynomial terms, and here are the second order fractional polynomial terms. And then lastly, we get the likelihood and link function. So it is a binomial likelihood with the complementary log link function, and the offset is logarithm of the length of the time interval. And this can be then modified as needed for different models. And then let's move to the last part, which is the model selection part. And we will look here a little bit, exploring the different models with R, and then testing the goodness of fit via AIC and visually checking. And here I also want to say what we are in the paper proposing as a model building strategy. So you do the model selection using this frequentist fixed effect NMA and generalized linear models framework for fitting the models, and you can very quickly explore many models with that one. And in fixed effect model setting, the Bayesian model select models with wake priors or non-informative priors, and the frequentist NMA results agree. So we should end up on selecting the same model, but the benefits with this is that the models are much faster to fit. But then in the second piece, which I will not go detail here, but that is then putting the best model into Bayesian framework and fit there the fixed and random effects NMA, and that gives really the flexibility then also for the characterization of the uncertainty, so for derived parameters, cost effectiveness models, and so on. And this is actually the same as is done in the paper of Janssen, so the first paper on the fractional polynomials. So at the end we end up doing the same analysis. It's the model selection which is done a little bit differently. Yes, okay, but so let's talk about the model selection via an application, which is the same application. So it's modeling survival over time for different cancer therapies, network of 4 treatments and 7 studies. And if we look at this figure is all the 7 studies and their Kaplan-Meier curves calculated from the binomial data, and just looking at this, we can of course see that there are some studies where the the survival curves are crossing, or there might be perhaps a delayed effect or so on. But anyway, it's clear that the proportional hazard assumption does not hold, at least not for all of the studies and treatments. Then let's go to the R implementation. And the 9 models that were showed in the first part. Here is how one can give them as a list in R. So you just specify as a list all of the models and what are the time-dependent functions. Time-dependent functions, and then you can loop over the models and fit all of them. I don't have this in my slides, but it's all in the appendix code, so one can try it out. And I will discuss it more in the next slide. But here we also then specify separately G1, G2, F1, F2. Which we use the G functions always for study and F for treatment. For most of the cases, they are the same. So the time-dependent function is always same, but with proportional hazards with viable baseline, we have a time-dependent effect for the study. So basically for the baseline, but the treatment effects are proportional. So that's the reason why we have two different functions. And then R code specifying the different models. Yeah, so here is the same example as earlier, which we looked in detail, which was the second order fractional polynomial with P1 equal to -1 and P2 equal to 1. And how would you give these then as a list of function? And here is what I just also already explained, that you can then also give proportional hazard with baseline survival by specifying the study specific event time functions differently than the treatment specific. Okay, yes. So once these are all run for all models run for the data, we can start to select the best fitting model, and the first thing which is usually look, it's the AIC, Kaikes information criteria, which is actually in fixed effect model case, and then the Bayesian with uninformative priors. This is equal to the DIC in the Bayesian framework, and the values should closely match. And if we look at the AICs for different models, well, obviously the exponential proportional hazard has the highest AIC, so it does not have the have the best fit. But then looking which models have the lowest, so which might have the best fit, we have second order fractional polynomial, and then we have piecewise exponential with 2 cut points at 2 and at 12. And highlighting these from the hazard ratios over time figure. So we have one figure here, which is the second order fractional polynomial model, and then we have the piecewise exponential, and for treatments 3 and 4, obviously a little bit different shapes, but not too much. But then for treatment 2 versus treatment 1, the shape is quite different. So with the fractional polynomial, we kind of, it's first increasing and then decreasing, whereas piecewise exponential, it's also increasing at the end. However, what is also concerning is that it's the confidence intervals are exploding wide, and then when we dive more into details, one way to check the model fit is also to check the estimated hazards. I mean the model estimated hazards against the observed hazards. And in these figures, I'm doing it for the 2 best fitting models. So I have the piecewise exponential model, and then I have the second order fractional polynomial model, and each row is each study, and then we are plotting the treatments which are in each study. And if the model is fitting very good, then we of course expect the observed hazards to be very close to the modeled ones, or estimated from the model, and that is a good comparison to do that, how much deviations you have with the residuals. But then what is here, for example, if one looks the study 2, one can see that okay, the piecewise exponential model is fitting perfectly for the last study points, whereas fractional polynomial, the function doesn't go there. But this is due because we don't have enough data in the last piece for these treatments, which of course tells that maybe or that we shouldn't probably be doing the piecewise exponential model because we don't have long enough data for these treatments. But yeah, so generally checking what is happening with the residuals is another good way to check the model fit. Okay, and this was now the part of the presentation and learning. So I hope that there is I was able to give kind of basic understanding how we do the parameterization, and what can we model with fractional polynomials, so different functional shapes, and piecewise exponential model, which gives us proportional hazard in different pieces, and then linking how to fit these models with R in the fixed effect setting, and how the model parameters link to the R code, and last but not least, how to do the model selection part. Regarding the R codes, so the paper supplement materials are all available in the supplementary materials, and here is a link to them. And then there has been also last year published in research synthesis methods NMA SERV and R-Shiny application for network meta-analysis based on survival data. This is not published by me, and it has many different methods, but also these are implemented there if one wants to explore different models. Yes, and at this point, thanks for all the audience, everyone who registered and joined, and then also I would like to thank the special interest groups of EFSPI and PSI for giving this opportunity to present and to exchange knowledge on these topics. Here are some references, and yes, then we will conclude with the same slide advertising our HTA special interest group.
Excellent. Thanks so much, Anna, for that presentation. And it's not an easy topic to talk about because it's quite complicated, but I think you did a smashing job. So thank you so much for doing that. What we're going to do now is open up the Q&A. So we have, as I mentioned at the start of the session, we've had a lot of submitted questions in advance, which is fantastic. So what we're going to do is kind of talk through those, and then if we do have time at the end, we'll see if we can take any other ones through the question box. Maybe to get us started, Anna, so these methods, I think they're familiar to a lot of us, but we don't necessarily have a good idea of how often they're used, or if they should be used more, if they should be used less. I guess linked to that, how well accepted they are. So first question would be, do you have any thoughts on how often the methods are used, and do you think they should be used more, or any other thoughts on that?
Yeah, I mean, obviously they have been used in HTA submission, and they are also given in some HTA guidances and so on, and I had a look also, like, because I haven't been applying these for a while myself, but a little bit about the reference, like citations for Janssen paper and for my own, and then there are a lot of applications. Then the question that how often they should be used, or if they should be used more than they are, that's difficult to say. I mean, if they bring value, I mean, if we have a non-proportional hazard setting, and if we can somehow better explain the data and the clinical benefit and how it changes over time, then of course, but this all comes to the generally like what is the best fitting model, and of course there are some new ways nowadays as well, and getting, yeah.
Thanks, Anna. I think it's probably fair to say that they're complicated, like you said at the beginning. I think when we were planning this webinar, you shared the anecdote of when you were asked to fit these models for the first time, and your initial thought was, Ah, this is really complicated. So I think,
yes,
I guess the acceptability of the methods, if you'd agree, is probably a little bit of a function of how complicated they are. And so there's maybe select cases where they really are necessary, and perhaps others where it might be harder to justify.
Yeah, yeah.
Great. Okay. And then moving on from that point, I guess a key thing that I think would be helpful for people is maybe from your experience using these models, what do you think are the biggest problems or limitations of these types of methods? So where do they maybe go wrong most often?
Yes, so then I think it's, I mean, what is challenging always is, I mean, at the end we want to extrapolate the survival. And how well can we do it with the models? Sometimes probably better, sometimes worse, but that still remains like can we extrapolate from the models, and is the extrapolation kind of reliable? Then one thing which I found when I was working a lot hands-on with this is that I mean, when you calculate the hazard ratios, those are fixed. But then when you go back to the survival functions, you of course need to select some baseline survival, where you then add for the treatments, and there I feel sometimes, and it's not completely clear to me how it, but like the selection of the baseline treatment can actually have quite an effect how the like how the differences look as well. So this is something that is also, and then of course this fact that it is complicated to implement. I mean, and there are many steps where it's complicated. So the data extraction and derivation, then fitting the models and reporting and so on. So one can. I mean, it requires heavy coding and validation and so on, and then really understanding that if you know if there is something weird going, where is it coming from, and so on. Yeah.
Thanks, Anna. I think certainly when you sort of outlined all the steps that are involved, it's quite an adventure, isn't it? So you kind of go from this data, you have to do the grouped data, and then you have to set up the models. And of course, one thing I'm going to move us on to in a second is about model selection. But you talk about the kind of frequentis fitting of those models, find your best fitting, and then focus in on those. And I think certainly that's quite a long process, but a necessary one to have these models work in practice. Yeah. So linked to that, what views do you have on, I guess, the two extremes of you must find the best model, and that's the right thing to do, and that should be the one we aspire to find, versus finding a group of models, and maybe there's a series of options or a range of different models that might provide different estimates. What do you think is the thing people should be doing? Should we be trying to find the best model? Because you highlighted that you looked at 9 different models. So should we be trying to find one, or should we be trying to find a small number of good fitting models and then allow there to be some uncertainty there?
Yeah, I think there is like, and then I need to say here as a disclaimer that I'm not so familiar with the health economic modeling, where then of course these models serve as an input there, and then you select, I guess, one model, but also there, yeah. So but purely from statistical perspective, then I think it's good to kind of, I mean, it's great if there is a model which seems to fit well for the for the whole network, but then you might wanna, I mean, it's also important to do this evaluation that you compare the models, that you see that what might be. And then the clinical interpretability is of course one thing as well, that for example, the piecewise exponential could be quite simple to explain that, okay, we have a delayed treatment effect of 3 months, and then it makes to have a cut point there, and then you can talk about the hazard ratios in the first piece and second piece, and so on. Yeah, and then the third point, perhaps that I guess it also depends what is the treatment that is the most interesting, because if you have in your network some treatments which are not so interesting anymore for the research question, well, then you also discuss, of course, do you need them or not? Sometimes you need for some, getting the network and so on. But that the actual decision at stage that you would have the best model to kind of answer those questions.
Completely agree, Anna, especially that your last point there, that was really clear based on the hazard ratio plots you shared, where if you chose that best fitting fractional polynomial, it implies towards the end a very different long-term effect when you compared it to that piecewise exponential model. So I guess it's a time and a place situation, isn't it? So sometimes maybe there is an obvious winner of a model, and then maybe in other cases it isn't. From a statistically enthusiastic health economist perspective, which would be how I'd identify, I think that the challenge with putting this into a health economic model is that then it's one piece of an even bigger puzzle. And so you've got all sorts of things to consider there. And I guess as well, if it's a Bayesian model, you have to think about how to capture that properly within things like a PSA, so probabilistic sensitivity analysis. But that's I guess for us to worry about, Anna. So,
yes.
It's very kind of you to think of us, and I think that's another challenge, I suppose, with the use of these models. I was going to ask as well, so we talked a bit around model selection, and I think you helpfully outlined the role of AIC for helping to slim down that number of models that you fitted in a frequentist framework, and then focusing on maybe the best ones in a Bayesian framework. You also, as well, I think you flagged the residuals, I think, was on that towards the end. Is there anything else that you think people should do if they were trying to defend a particular model over another?
Yeah, so I think it's really what was already mentioned, this extrapolation and prediction, and how do they behave there, and then the discussion with our clinical colleagues that what is kind of what supports the clinical feasibility, what is plausible in that sense. That I would say.
And I think as well, you mentioned earlier around if there's a plausible time point, then naturally something like a piecewise model feels correct. It feels like a sensible choice. And I think there'll be perhaps people watching that have seen analysts' choice of a cut point is simultaneously a good and a bad thing. I think people don't sometimes enjoy the concept of the analyst determining when something happens, but equally, if there's a good reason to do it, it can be helpful. So I think what's neat about the methods you talked about today is you can show a piecewise exponential model where there's an element of choice there in terms of where you place that cut point. And I guess in theory, people could change where that cut point is and run it again.
Yeah, but this actually brings another additional point, which is good. I mean, generally with these ones, also like the pre-specification, you know, the pre-specification of the models that we fit, and how do we select is of course important as well if we wanna be more credible on you know that what was the, and transparency is important there.
Absolutely,
yeah.
Absolutely. Thank you. So we've had actually, I think, this question a few times. And I think it's probably an important one, which is towards the start of the presentation, you showed the original formula, which was that crazy complicated one, and you mentioned it was from your own Janssen's paper back in 2011. I think the audience would be keen to understand if there are any differences in that original specification of the fractional polynomial model and the model that you've implemented. So are they the same model or are they different?
Yes. So and this is a really good question, and I think it's important to so obviously there are differences in the parameterization, and then that we do frequentist or Bayesian model fitting. But then for the overall process, it's and then for the decision making, we should end up with the same result. And because we use this in my paper, we use this hybrid approach that we do just the model selection with the frequentist. But what are the key differences? So I use this different parameterization. And in one of the references, there is the paper by Pifo, where this parameterization is explained just in a standard NMA, and these models, when it's, so let's now leave the Bayesian aside, just the models like the parameterization of the linear predictor. So if you have a fixed effect model, this R-based parameterization in a way that it's done in the Pifo's paper, and the baseline contract parameterization, which is quite often used in fixed effect case, they are exactly the same. So it's just, and this is also discussed in my paper or in our paper, and then there is of course this Bayesian versus frequentist model fitting. And in fixed effect case, the differences are, I mean, they should in fixed effect case with unitary priors, it yields to similar results. So those are kind of the differences that we use this different parameterization and model fitting. But then on the other hand, the final model is run using Bayesian analysis and similar model specification.
Great, thanks, Anna. And I think we were just talking, weren't we, before we started, about how depending sometimes on things like computer processors, software packages, all sorts of other things, you can get these small discrepancies in the Bayesian models. I think a number of chains, blah blah blah.
Yes,
but it's helpful with your analysis because it builds on a set of frequentist models. It can be perhaps more easily verified that those are the same specification.
Yeah. And there was this one specific questions from the audience, which was about why do the results, the parameter estimates, why are they different in our paper versus the original Janssen paper, and obviously I cannot repeat the analysis in the other paper, but I think it's likely because the number of simulations are, I think there was like 30,000 MCMC in Janssen, and I had like 200,000. So and here is of course the time has also, you know, like 2011 versus 2020, the Bayesian has also approached. But what I think is important comparison, that does the Bayesian fixed effect model match with the frequentist frequent is, and that is the good comparison to make. And those are, I mean, there are some very small numerical differences, but they do match generally. Yeah.
Excellent. That's great. Thanks, Anna. So we're very closely encroaching on the end of time, so I'm going to see if I can squeeze a couple more questions in. And again, sorry to those who've submitted them. We're probably not going to get through all of them, but it's great to have so many. We're glad it's been engaging enough for you to stick around. What we wanted to just check, I guess, briefly, Anna, for your thinking is the piecewise approach. You mentioned there's obviously the choice of where to put a certain cut point, but there's another choice of how many cut points you use, and you looked at a model that had one cut point, and then you looked at another one that had two cut points. So do you have any particular views on how many cut points is maybe a sensible upper limit, or is that maybe case by case?
Yes, maybe case by case, but then I would say that there is really this clinical plausibility is also important, and I then for example, two cut points could be something which is, we can have this delayed effect, and then there can be happening something later. Then if we start to have many more, then it's can we explain why we think that many more are needed? Yeah.
And then I think we've got time for one last question, and so it's a big one, and there might not be a short answer, but let's see how we go. So quite a few of the questions submitted have asked about some of these newer methodologies. So there's the multi-level network meta-regression work that has come out more recently, use of M-splines in the serve-extrap package by Jackson, and there's probably others. So as a general question, do you have any particular views on how those newer methods could or should be used, how they might compare to this method? Anything that you think you want to share?
Yes, I think these were great questions. So I was very pleasure, and here I haven't been applying so much with the new newer functions myself, but I did look into it. Then I think there is like, for example, because in some sense it's like the time-dependent covariates. I assume that you can be putting them, in the GLM framework, or then you can have the, and it can also be that it's so the same fractional polynomial model can be implemented on both, and then I guess it's matter of preference that which you are used to use. And then the question of splines, which was actually coming quite a few times, and this is something that I was looking that actually could you with this kind of model fitting also do the NMA with splines, and I think it's probably doable. But then I would need to do it in practice. So this might be something to consider as well. But then really this question that is it spline fractionals, polynomials, or piecewise exponential? It's or something else even.
And hopefully this training can help people with at least the starting point before we get too crazy with all the methods, but that's really helpful. And so I know we've run over ever so slightly, but thank you all for your attention and for participating today. There'll be a follow-up email about if you have any other questions. So if we've missed them, please do put them there. We'll do our best to answer the ones that we can. And otherwise, I'll maybe let Anna have the last words, and then we'll close off.
Yeah, so it was a great pleasure being here, and I hope I hope this provided some clarity about the, you know, like really the fundamentals there, and like I said, all the questions are also welcome through the email or LinkedIn or so on. So and thanks for those that were there, because it was a good learning exercise.
Great. So with that, we'll close the webinar, and thank you all, and enjoy the rest of your day.